After completing his graduation from a prestigious pharmacy college, Ramesh joined a vaccine manufacturing facility as a trainee operator. Ramesh had just completed his training and first day stepping into the critical environment, an aseptic manufacturing area where sterile pharmaceutical products were formulated, filled, and sealed. He had heard many stories from his colleagues about the precision and attention to detail required to work in such environments. As he suited up, Ramesh understood that his actions could be the difference between a successful batch and a contamination event that might compromise patient safety. His mentor, John, a seasoned aseptic operator with years of experience ensuring sterility in the manufacturing process, would be responsible for overseeing a media fill, a process simulation that tests the sterility of the manufacturing line without using the actual product. Instead, it uses a microbiological growth medium, which, if exposed to contaminants, will reveal microbial growth during incubation. "Everything you do here is about protecting the product," John said, his voice clear through the speaker in his sterile suit helmet. "We use these simulations to ensure that nothing we do, no movement or intervention, risks contamination." Ramesh nodded, fully aware of the task's importance. He had spent weeks learning about aseptic techniques and the intricacies of process simulations, how to properly gown, sanitize surfaces, and minimize interventions that could compromise sterility. However, this was his first hands-on experience with an actual media fill. The sterile filling area was a pristine space, kept at ISO 5 conditions, meaning fewer than 100 particles per cubic foot of air were allowed. The filling machine stood ready, its polished surfaces gleaming under the bright overhead lights. The sterile media, soybean casein digest medium, had been prepared and sterilized, now ready to be filled into the pre-sterilized vials. "The environment is our first line of defense," John reminded him. "We rely on environmental monitoring to ensure it stays sterile, clean rooms, HEPA filtration, air velocity, temperature, and pressure differentials. But it's our job to make sure we maintain aseptic conditions by minimizing any potential contamination sources." Ramesh remembered the countless sessions on environmental monitoring programs. The facility's environmental conditions were continuously monitored, with sampling points spread throughout the critical areas. Any deviation from acceptable levels of microbial or particulate contamination would trigger an investigation, a precaution to prevent product contamination during routine production or simulations. The goal of today's media fill was to simulate the entire aseptic filling process under normal operating conditions. The filling process had already undergone several risk assessments to identify potential weak points, places where interventions could introduce contamination. "Remember, Ramesh," John said as he checked the machine, "the key to aseptic process simulations is that they challenge the system. We're not just going through the motions, we're actively testing the process, equipment, and even ourselves." The process began smoothly, with the machine filling vials at its predetermined speed. Ramesh kept his eyes on the vials as they moved down the line, each one sealed with a stopper as it left the filling station. "Now, let's talk about interventions," John said. "There are two types: inherent interventions that are part of the process and corrective interventions that we only do when something goes wrong. Today, we'll be simulating both." Inherent interventions were expected. During the fill, Ramesh had to replenish the stoppers and adjust the weight as part of the normal process. But corrective interventions, like clearing a jam or adjusting sensors, introduced more risk. Each time Ramesh or John touched the equipment, they followed strict protocols to maintain sterility. Every movement was slow, calculated, and deliberate. "During a media fill, we simulate worst-case scenarios," John explained. "We purposely introduce a little more risk, like performing an intervention we might usually avoid, just to test how well the process holds up. We even simulate shift changes and breaks, so multiple personnel changes are covered." John's words echoed a key principle Ramesh had learned: the concept of worst-case scenarios. Every process simulation pushed the boundaries of normal operating conditions, filling the largest container size at the highest speed or executing interventions with the maximum number of personnel present. These simulations ensured that even under the most challenging conditions, the sterile process would remain uncompromised. After the fill was complete, Ramesh and John inspected the filled vials, checking for any visible issues with the seals or closures. The vials were then incubated, where they would sit for several days under controlled conditions. Any contamination in the batch would cause microbial growth, revealing itself during the post-incubation inspection. We'll review the environmental monitoring data, too, John said. If we see any environmental excursions, like a spike in microbial counts, it could explain contamination in the media fill. But the goal is no contamination at all. If even one vial shows growth, we have to investigate. The investigation would be thorough, covering every step of the process. Was the equipment properly sterilized? Was there an unplanned intervention? Could there have been an unnoticed breach in aseptic technique? These questions would help identify the root cause of contamination, leading to corrective actions to prevent future issues. Personnel qualification was a major part of aseptic processing. John told Ramesh how each operator had to pass initial gowning and technique certification before they could even set foot in the clean room. Periodic requalification ensured that everyone remained proficient in their aseptic skills. Even our movements matter, John said, demonstrating how to enter and exit the filling line without disrupting the clean airflow. Slow, smooth movements create fewer particles, reducing the chance of contamination. Ramesh followed his lead, carefully maintaining his sterile gloves and gown as they completed the fill. He knew that personnel were the most significant source of contamination in aseptic environments, so every operator's actions had to be impeccable. As they completed the media fill, Ramesh understood the weight of his responsibility. He was part of a process designed to protect patients who relied on sterile, safe pharmaceutical products. Each step in the process, from environmental monitoring to aseptic technique, was another layer of protection, ensuring that nothing compromised the sterility of the final product. In the weeks that followed, Ramesh would monitor the results of the media fill and participate in the root cause analysis if any contamination was found. But today, he walked away from his first hands-on experience with a new appreciation for the precision and care required in sterile manufacturing. It's not just about following procedures, John said as they removed their gowns. It's about understanding why we do it. At the end of the day, the patient's safety is in our hands.